Assalamualaikum warahmatullahi wabarakatuh. We are from Group 3 and we will present about genetically engineered aubergine.
Assalamualaikum warahmatullahi wabarakatuh. My name is Mohamad Afiq bin Mohamad Khairi, 1170350. For the introduction, eggplant, Solanum melongena, or brinjal, called in Bangladesh and India, is one of the most important, inexpensive, and popular vegetables in Bangladesh. The biggest constraints to eggplant productions are chronic and widespread infestations by the brinjal fruit and shoot borer BFSB. The most serious economic damage caused by BFSB is to the fruit, because the holes, feeding tunnels, and larval excrement may make the fruit unmarketable and unfit for human consumption. Studies have found that the farmers spray from as few as 23 times to as much as 140 times per season in order to fight this pest. This practice is unhealthy for consumers, farmers, and the environment, and it is also expensive to farmers.
Assalamualaikum, I'm Shakirah with matric number 1171473. We present about mechanism of genetically modified aubergine. The genetically modified aubergine is a suite of transgenic aubergine, also known as eggplant or brinjal. A transgenic plant contains one or more genes that have been added from another type of plant or animal. A transgene is a gene that has been transferred naturally or by any of a number genetic engineering techniques from one organism to another. This non-native segment of DNA may either retain the ability to produce RNA or protein in the transgenic organism or alter the normal function of the transgenic organism genetic code. Genetically modified aubergine created by inserting a crystal protein gene, which is Cry1AC, from the soil bacterium Bacillus thuringiensis into the genome of various aubergine cultivars. The insertion of gene into the aubergine plant is accomplished using agrobacterium-mediated genetic transformation along with other genetic elements such as promoters, terminators, and an antibiotic-resistant marker gene. The Bacillus thuringiensis aubergine has been developed to give resistance against Lepidopteran insects, particularly the aubergine fruits and shoot borer, which is Mucinodes urbalis. The fruit and shoot borer FSB, known as Mucinodes urbalis, is the most devastating and troublesome insect in aubergine crop. Bacillus thuringiensis aubergine was approved for commercial release in Bangladesh in 2013. How does Bacillus thuringiensis aubergine control fruit and shoot borer? This diagram summarizes how Bacillus thuringiensis aubergine control fruit and shoot borer. For the next part, I will pass to Danial.
Assalamualaikum, and I am Mohamad Danial. So today I will present about the mechanism of the genetic modified brinjal. First and foremost, Mahi company licensed and use the Cry1AC gene that is obtained from Monsanto and two supporting genes, which are MPTL and AAD for these processes. The Cry1AC gene is under the transcriptional control of an enhanced cauliflower mosaic virus 35S promoter, which ensures the gene is expressed in all the brinjal tissue throughout its complete life cycle. MPTL and AAD are selectable marker genes, which MPTL is used to identify transgenic plants from non-transgenic, and AAD is used to identify the transformed bacteria used during the development of the construct. AAD contains a bacterial promoter and it is not expressed in the brinjal. The completed construct was then inserted into young cotyledons from the brinjal plants using an agrobacterium-mediated technique. Agrobacterium naturally inserts DNA into plants from its TI plasmid, and scientists use this to insert genes of interest into various plants. The transformed plants were then regenerated and analyzed for the presence of the gene through Southern blotting. This plant progeny also was analyzed to identify the lines segregating in a Mendelian fashion, as we usually know. Thank you.
Okay, my name is Farah and now I will talk about advantages. First, it will increase the resistance to ESFP. Aubergine, shrimp and fruit borer ESFP is a major insect pest of aubergine. When the CRY1 AHA gene is expressed, it will give a result in high ESFP level mortality in the shoot, as well as fruit tissue of the transformed aubergine lines. Such transgenic aubergine line possessing resistance to the ESFP could reduce the dependency on pesticide use for ESFP control. Next, it will also reduce the usage of chemical pesticides. In Bangladesh, the farmers were able to reduce the number of pesticide application by 33.6% compared to the control group. The quantity of pesticide used also decreased, while the toxicity of pesticide declined by 42% overall. Farmers growing GMO aubergine and who had pre-existing chronic condition were 11.5% points less likely to report the symptom of pesticide poisoning. Thus, it leads to a more safer environment and aubergine plant. Next, I will pass to Hanisah.
My name is Hanisah Binti Mohamad Ikhiwan. I will continue the presentation on advantage of GMO aubergine. So the next advantage is increased profit and productivity. It is proven that GMO aubergine produce more crop than normal aubergine. So when cultivated in greenhouse and open field, significant increase of yield was observed. In addition, aubergine also used less pesticide compared to non-GMO. In short, the profit escalated as the production increased and the pesticide usage would decline. In addition, the aubergine also is high in nutrient content. It is a good source of fiber and water and also rich in antioxidant. Aubergine also is high in vitamins and minerals.
Okay, next we continue with the disadvantage and issue of BT brinjal. So firstly, high cost of BT brinjal seeds. So the cost of BT brinjal seeds are higher compared to conventional brinjal seeds. This is due to the complex process and also research done to produce pest-resistant seed. Okay, second, uncertain impact on consumer health. So genetically modified crops normally will synthesize new protein that can express the desired trait of interest. However, this crop normally linked to the issues such as have bad side effect on the health of consumer due to the gene modification and also the production of toxic. Furthermore, there is no scientific consent and safety assessment have been reported regarding this BT brinjal. Okay, besides that, the establishment of BT brinjal also can develop pest resistance against the BT brinjal. So BT brinjal is a transgenic crop that have the ability to produce secondary metabolites such as toxic product for resistance toward pests. However, sooner or later, the pest might develop resistance against the toxic product produced by the BT brinjals. So the same case was happened to BT cotton. And besides that, the production of toxic also may kill off other insects, which is important for ecosystems. For example, like insect that is responsible for pollination process.
I will proceed on the disadvantage and issue of BT brinjal. So the fourth issue is on the antibiotic resistance. In BT brinjal, it consists of two antibiotic marker genes. The first one is NPT2, which code for neomycin phosphotransferase 2 that is resistant towards kanamycin antibiotic. The second gene is AAD, which is resistant towards the streptomycin antibiotic. So this antibiotic resistant gene can be transferred to bacteria in human or animal gastrointestinal tract, or even worse, in pathogenic bacteria. So it will make the pathogenic bacteria resistant toward the antibiotic and can grow in antibiotic medium. The fifth issue of Bt brinjal is the ecosystem imbalance. Bt brinjal that contain insect-resistant gene can affect the environment by harming the insect, because even the non-target insect can be threatened if they consume the toxin directly from the pollen or plant debris. So this will lead to imbalance of ecosystem because it will reduce the beneficial insect that can act as pest control.
As a conclusion, genetic engineering is a scientific advancement that has contributed to biotechnology advance. Genetically engineered crops that provide insect and disease protection or herbicide resistance are essential instruments that enhance a strategy for diversified pest management and could increase productivity. It also benefits mankind when used for purposes such as increasing the availability and quality of food. However, there will also present challenges that need to be addressed where it is safe enough for genetic engineering. This is related to ethical issue. Abu Hurairah Abdul Rahman bin Sakh reported that the Messenger of Allah said, "Do not be envious of one another, do not artificially inflate prices against one another, do not hate one another, do not shun one another, and do not undercut one another in business transaction, and be as fellow brothers and servants of Allah." Narrated by Muslim. From this hadith, the Shakirah law is applied, which harm should not be inflicted nor reciprocate. Most verses related to modern biotechnology in Al-Quran have only mentioned in the basic rules in Islam, while quotation from the hadith describe how Muslims should conduct their life and guide Muslims to behave themselves toward being good person and to be wary that unethical behavior result in negative impact. Thus, risks and benefit of genetic engineering must be analyzed in terms of ethics and safety. Any harm caused by genetically modified organisms in the long term or the short term on any of Allah's creations is prohibited in Islam based on the source of ethics of Al-Quran and Al-Hadith.
That's all from us. Thank you.